Testing on, testing on, testing on, testing on.
Right. Good evening, everyone, both online students and physical students. So we are back in the class. We will be discussing today about mitosis. We started discussing cell cycle, right? So I'm just going through the cell cycle one more time, refreshing the memory that we were discussing. I'll be asking few questions from the ones who were present in the class. Are the absent ones, just open the book and keep. And if you can give answers, your answers also will come to the class. Right. So cell cycle. It's a cycle, no? So it's talking about mitosis, right? Right. So starting from here until this point, this is the longest phase of the cell cycle. What do we call it, Pute? Interphase. Ah, so interphase, I'm going to divide into how many phases? 3 phases. Good. So here it is. What are the 3 phases? G1, S, G2. What's the meaning of G1? Gap 1. S, synthesis. Then G2 means gap 2. Another. So I'm going to discuss what are the events happening in G1 SG2 one more time. Another. Right. So G1 phase. What are the things happening here? Synthesis of new proteins. Hi, good evening. Hi. Synthesis of new proteins and what else? Production of new cellular organelles. What's actually happening here? The cell is going to get ready for division, so that G1 is G2, the cell is preparing, and by making everything, doubling the number of chromosomes and doubling the number of organelles, and everything is being prepared before it enter to the division phase, right? Alright, then S phase is special one. What's happening in this phase? DNA replication. What's the meaning of DNA replication, children? Making same copies of DNA. That means DNA is copied and making genetically similar another copy. Right. So the DNA sequence is repeated in the next one. Right. So then next one, histone protein synthesis. What else? DNA is winding around histone proteins. It is like this, you know, DNA is something like this, no? Correct? So what is the shape? What is the structure we called? DNA double helix. This one is going to wind around 8 molecules of histone proteins. Likewise, there are many 8 molecules of histone proteins. I'll draw a few here to show you only. So this DNA molecule is winding around this histone protein, right? What's the reason for winding like this, children? What's the reason for winding around this histone proteins? Do you have any idea? Just give me a guess. Online ones also can give. Waiting for your answers too, Imana. There's another one, new one, Samiksha, no? Okay, right. Samiksha, you're also welcome to the class, right? Tell me what's the reason for winding this one? Otherwise, DNA would have been in the nucleus without winding around histone proteins. So why is winding? No. What's the reason for winding? Do you have any idea? I'm not laughing at your answers. Just give me an idea. DNA molecule is very lengthy molecule, isn't it? So can we pack a very lengthy molecule in a small space? No, it's difficult, right? So that we are winding around these histone proteins and reducing the size of the DNA molecule and making sure that everything is put inside a small amount of that nucleus size. Okay, understood now? So once the DNA is wound around histone proteins are only found in whom? Eukaryotes only. We call this as what? Chromosomes. Here in the interface, the chromosomes look like network of chromatin. What is network of chromatin, children? These chromosomes are loosely packed. These are loosely arranged, not a compact structure. Now, this is like a Maggi noodles. It's like diffuse mass. Okay, understood now? Hi. So, this is how it is happening. So, I was explaining this one. And what is happening in the G2 phase? Protein synthesis, production of cellular organelles, and duplication of centrosomes. Actually, the centriole is the one which is duplicating. Centrosome is a region where the centrioles are present near the nucleus. Okay. Hi. Just will give you a minute. Just go through this one more time before I move into the next section. And okay. Right. So now moving to the M phase, which is happening after the interphase. I have done this section. Those who came today, just to go through the recordings or ask from a friend. Hurry. So that I'm erasing this part to get some space for the next section. Hurry. So here this we call is as M phase. M phase is having two major phases. What are the two major phases? Mitosis and cytokinesis. Here it is mitosis up to here, and here it is cytokinesis. Right. Then mitosis is having how many phases? 5 major phases are there. 1, 2, 4, 5. Right. Now you guys tell me what are the 5 major phases in mitosis? Prophase. What else? Prometaphase. Then metaphase. Anaphase. And telophase. I told you that there's nothing as called prometaphase. Since the prophase is very big, they have divided it into another one. So it's a new addition. It is not taught in the meiosis lesson. Okay? So just for this one, you will be learning about prometaphase. Up to this one, okay? Let's define the cell cycle also. What's the definition of cell cycle? The sequence of events. I'm breaking the definition into parts, right? Let's see how many parts are there. The sequence of events occurs in a life cycle of a cell. No? How they have given? Tell me exactly. Hmm? Harine? Life of a cell. Then from the end of one cell division until the end of another cell division. Now, okay? So, isn't it easy to break a big definition into pieces to study? That's how we should study, right? So, let's say you are going to study this one more time. You will be putting numbers here 1, 2, 3, 4 parts are there. You are taking a piece of paper, you are writing and you are checking all the 4 parts are there or not. Remember very well, in the exam, this is the one particular marking point. If you have missed one of these wordings here, you are not going to score the mark here. I told you that in the structure they say the marking is very strict. They are considering each and everything to give 1 mark. So that you have to be very careful when the marking is like that. I have been to marking, so I know that even if you feel like this child has understood and written it in his or her language, but still we can't give the marks because the exact wording which is expected in the book version is not given there, right? It's very bad also in a way, but still, if you go through the marking scheme carefully, the professor's wordings are really meaningful. So that we can't give marks for the exceptional answers, right? Okay, understood. So I went marking last time. This time also I will be going. So in that sense, if the marking is started, sometimes there may be not in weekends. Sometimes it's happening, but some days they will be requesting Saturdays also. So if so, sometimes the classes will be online only at that particular time period. I'll let you know about that thing that is coming up after the exam. No? So I'm telling it earlier. So this time also I'll be going marking. It is important for you guys only. Hurry. So what I was telling is what's happening in the marking. So this way it's easy for you to study when you are studying now, so that you won't miss any part of the a big sentence. Okay, so get used to this method, right? Okay. So now you go to the definition that you wrote in the book and put numbers and come. Take a pencil and put numbers. Others, those who don't have, just write this and keep. Cell cycle. Definition of cell cycle. It's a cycle now. Remember very well. Where you start, you have to stop. Here, from the end of one cell division until the end of one cell division. Harder. Don't tell that until the beginning of another cell division. Okay? If you start from the beginning, you have to stop at that place. You can say like this also. From the beginning of one cell division until the beginning of another cell division. Harder? Did we draw only prophase? We didn't write anything on prophase, no? Here you don't write cell cycle definition after the diagram. We are continuing the lesson. I told to write only the ones who came today. We wrote the definition early. You can remember. In the question, no, question, yeah, that's right. Hi. Okay, right. Now do you remember the cell which was at interface? I'll be quickly drawing cell which was at interface also. So here is the cell. There is a prominent nucleus. Nuclear envelope is there. And the nucleolus is visible. Their DNA is visible as what? Network of chromatins like this. And the centrioles are there like this. Alright? Okay. So this is the cell which is at interface. That means the getting ready phase. So cell is next will be going to the prophase, right? So what's happening in the prophase, children? The network of chromatin condenses and forms what? Chromosomes. Chromosomes look like what? Chromosomes with two sister chromatids. Harder. Let's draw that onto. Chromosome is having two sister chromatids. Earlier, it was network of chromatins, but inside that actual one which is there is this. This is the single chromosome. Harder. Okay. So if I give you another reminder again about DNA number and chromosome number, I told that here it is having how many DNA molecules? One DNA. How many chromosomes? One chromosomes. Correct? Then after the S phase, replication occurs. Replication makes similar DNA. Same copy actually, okay? One arm is having one DNA. The other arm is having another DNA. So here it is having two DNA and one chromosome. Correct? What do we call this part? Centromere. Not centrosome, right? Centromere. And what do we call these guys? Sister chromatids. And also here is some protein attached. This one, what do we call it as? Kinetochore protein. Harne? Hi. Do you guys want to draw this one? No? Hi. Then we will be moving to a cell which is at prophase. Right? So what's going to happen, children? You guys tell me what's happening here. Nuclear envelope is there, no? Nucleolus, what happened to the nucleolus? Nucleolus disappears, no? So there's no nucleolus hereafter. And the chromosomes now look like chromosomes with two sister chromatids. I told you not to draw like what? Not to draw like butterflies, no? Some are drawing like this. It's totally wrong. Why is that? These chromosomal arms are bound by a protein. What is the name of the protein? Cohesin protein. They are binding these chromosomal arms so that they are not separated. They are closer, right? Okay. Alright. So I'll be drawing few chromosomes over here. They are randomly found on the nucleus. Then what's going to happen to centrioles, Pute? They started to move towards the holes. And then making what structure? Aster and Spindle. Okay, no? Right. So that's done. So we are going to write what are the events happening in the prophase in the mitosis. Right. So we are writing like a list. Okay, I'm reading. You have to write. And if you have not done the picture, please keep some space for the picture and go. By the way, the ones who have paid the fees, please collect the tute from the office. Harad, don't forget to get it because there are some pictures also for the next lesson and this lesson. Harad, online ones also, please collect the paper and also another news. The test will be uh, given on not next week, week after. The test is up to mitosis, but mitosis is not included, right? Cell cycle things are not included. That means the main things which are included is cellular organelles, subcellular components, plasma membrane, those lessons, right? Because we stopped the previous lesson, previous one at which place? Up to nucleic acids we gave, right? So up to nucleic acids, the test is not getting, right? So the new ones, please study. You are given 2 weeks time. That means week after, not next week, week after you will be having the test. Okay. Online ones, I'm not going to share PDF to anyone. Please collect the tute from the class along with the tute. You will be given the paper also, but do not open until the exam date. So be honest and wait until the exam date. Okay. So this is the full paper having 50 MCQs, and second part is having 2-hour paper, which is having 4 structured essay and 2 essays, 2 mandatory essays. You can't select and write. You have to write, you know the procedure, no? Like similar way, similar pattern. You'll be having a standard test, right? So you can evaluate yourself. Don't be afraid. This is not a big thing. This is to evaluate yourself and get an idea where you are in the syllabus and where you need to improve. So please write that exam. Usually my exams are standard and hard. Okay. Right. So here it is. So you will have time now, get time and study really hard. Right. So I'll be erasing this part too. No, I started to give the note no. Right there, point wise. I'm reading what is given there in the book, but let's try together. Number 1, chromatin fibers get condensed by shortening and thickening and transformed into chromosomes. Transformed into chromosomes. Number 2, as a result, chromosomes will be visible. As a result, chromosomes will be visible through light microscope. Number 3, nucleoli get disappeared. Nucleoli get disappeared. Next one, chromosomes appear with chromosomes appear with chromosomes appear with 2 sister chromatids. Chromosomes appear with 2 sister chromatids attached at the centromere. Next one, Chromosomal arms of sister chromatids. Chromosomal arms of sister chromatids attached by special proteins called cohesin. Special protein called cohesin. Next one, the formation of mitotic spindle begins. The formation of mitotic spindle begins. Next one, spindle is formed by accumulated microtubule complex, which includes centrosomes, the spindle microtubules, and the aster. Next one, centrosomes move towards the opposite poles. Centrosomes move towards the opposite poles of the cell due to lengthening of microtubules between them. Next one, centrosomes or centrioles are not available in plant cells. Or centrioles are not available in plant cells. Next one, however, spindle is formed during cell division from accumulated microtubule complex. Hi. Now what phase is coming, Putin? Pro-meta phase. So we are going to quickly go into pro-meta phase. So now the nuclear envelope is fragmented. So you have to show that nuclear envelope is fragmented. And centrosomes are now it began to move into the poles in the previous one. So now they are almost closer to the pole. It looks like this now. Aster is there, spindle is there. So now the chromosomes are attached to spindle microtubules now, but they are not in the middle exactly. They will be randomly found here and there, right? So I'll be doing like this. There are some microtubules interacting both the poles also. Those are called non-kinetical microtubules. So we had 3 pairs of chromosomes there, isn't it? So from one side only, the chromosomes are attached to these spindle microtubules. One chromosome, I'll be drawing it here. Another one will draw somewhere here. The other one, let's say it's here. Okay, so this is how it looks like. Right, shall we draw? I'll label me while you're drawing. Get used to these diagrams, draw many times as possible, and few minutes time you should be able to draw all the diagrams in mitosis, right? So practice is important even in a diagram. Otherwise, what will happen, children? In the exam, it will take time to think and draw the diagram. You will lose the valuable time to write the essay. And draw bigger and nice diagrams so that it is convenient for you. Centrosome is the region where centrioles are present on the that is present located near the centrosome. Right, let's write the events also. Write number 1. Nuclear envelope fragments. Nuclear envelope fragments. Write in point form, right? Nuclear envelope fragments. This kind of things, picture says a lot. So if you can remember, you can write lot of sentences there. Next one, chromosomes get even more condensed. Chromosomes get even more condensed. Number 3, a special protein called kinetochore, a special protein called kinetochore attaches the sister chromatids of each chromosome at their centromere. At their centromere. Next one, some of the microtubules that attach to the kinetochore of the chromosomes, some of the microtubules that attach to the kinetochore of chromosomes move the chromosomes back and forth. Next one, microtubules which are not attached to the kinetochore interact with those from opposite poles. So by the way, children, do you know that the exam is coming up, the A-level exam is coming up November 25th. So the biology paper will be on 25th. Usually the biology paper part 1, part 2, 2 days now. So first day will be second paper usually. So first paper usually, second day, second paper, right? So there's no gap in between like in the other subjects. So you have to prepare and quickly write the second paper, right? So that means like you were taken once, 2 batches are here now. So that means 2024 ones was here when you are coming. 2025 ones also there. Now 2024 one will be over soon, so you will be also eventually upgraded, right? That means your concern like the attention that everything will be higher than before, right? So the first priority up to now 2024, then second priority is now 2025. Then third priority was you will be second priority list after the exam, right? So it will be like in 1 month time, let's say. So you have to be focused more, and you have to understand the workload is getting even more higher and higher when the time is passing. So your exam also will be on 2026 may be November. I'm not sure about the time duration, November, or it may be August because you started early than the other groups, right? So usually 2025 batch is a little bit slower, very slow than you guys. So that sometimes you'll be closer to 2025 batch in a few months time because they are going slow. Even in the schools, it's slow, right? Okay, right. So are you done? So read it one more time, prophase and prometaphase quickly. I'll be going to metaphase soon. Hey, that you two don't have a book? Have you, no? How up to which lesson you have finished? Your classes? Cellular organ is done. Mitosis started. Then okay, no? Can continue now. Hurry. You can get ready for the test also, right? So it will be evaluating you guys. How was the last test? Was it good? You guys did well, no? So here was good marks also from this class. Rashida. Right. So here it is. By the way, I couldn't ask your results now. Totally forgot it. So how many 9 is? You know? Oh, many 9 is, no? How many parties, no? Okay. Right. So what about online students? I would like to get your marks also. Some of Hirui actually called. Hirui is not here now. Hirui is also having 9 A's. And also the specialty here is I was very proud that A-level result the highest marks from Sangamita. No? So here all also now the highest mark from Sangamita, right? So I'm very proud of that. And here one student also coming to our class from Sangamita College, same batch. She is Hirui. 8 A's and 1 C. What about Imana? Anita කොහමද? Oh, 9 A's, no? You? Science C. That's fine. You can continue. Don't worry, right? You? 9 A's, no? Science A. 9 A's, no? You too? 8 A's. 1 C for what? Don't worry. What about you two? Science A. You? Science B. That's fine, right? So now the O level is just a basic qualification to get through this, to get selected to A levels, right? Harid, don't be overwhelmed by the 9As also. That is also some risk. If you are having 9As, you are thinking, I can do A levels really well. That's the false thing, okay? And the ones who are having with little bit less marks also don't get discouraged by the mark. Please understand your capacity and please do work well from the beginning itself. There may be real reasons why you didn't get a good mark because you maybe started work doing at the last moment. Last moment things anyhow won't work for A levels, okay? So sometimes some are getting 9As even if they are doing a little bit of going through the lessons and the last moment work. But A level is not like that. Throughout, you have to keep your head properly and you have to prepare well. That's the thing. Okay, that's what we are doing. We are helping you to get prepared. Harada. Okay, Hari. Hari. All, I would like to congratulate also. Hari. Especially we will take parties. Okay. Right. So here now the centrosomes are at the poles, right? Opposite to each other. There's no nuclear envelope now. So the chromosomes are going to get into the metaphase plate. That is, we call that equatorial plane also, exactly the middle of the cell. So that these are here now. Non-kinetical microtubules are also there. You have to continue with the same colors, otherwise the diagrams will be wrong. Now here at this point, you should understand that chromosomes are going to get attached to both the poles by microtubules. These are actually the cytoskeleton microtubules, right? These are involved in formation of Esten spindle. Okay, now? Hi, you guys start to draw. I'll label meanwhile. Okay, Mana, you also good. What about science? Pute, what are your names? I couldn't ask also. Saving you, Alindu. Who recommended the class? You just found and came. Okay. Do you know about me? So you are living close by. Kiribati. How long will it take to come here? 15 minutes. Then what about you? Same. You're from which school? You're Yoshida International. You? Ah. So how many are there in your national classes? Not everyone is doing science, right? You? Your schools are started, right? School, which lesson you are doing now? Same lesson. You didn't start yet. So after the results, you don't have A levels then. Then you must apply for schools, right? Just they have started taking applications. Please go to and may send the applications on time. What are the possible schools that you can apply? Yeah. Even the other schools also taking, but the thing is they are looking for 9 years. There's a big competition between 9 years people also. Sometimes even if you have 9 years also, they are not taking. They are looking for other qualifications also because that much of 9 years people are there. And there's another thing also happening in the schools nowadays. Students are not coming much, the government schools. But the thing is, they are not giving the chance to other people also. They are just registering at a school and not giving outsiders also. That's very tragic. Some school, the tuition teachers are also telling not to go to school. They are just going to tuition classes during the daytime and they don't know the value of the school also. Okay, Mana, don't worry that you can do science and still, but you have to concern on what you are doing, okay? What about maths, Mana? Samiksha, C for what? You guys are not changing the schools, right? Some don't have schools, ne? You have to apply, ne? Changing. You're looking for its school. DMS. Yeah, it's close by. Nice. You will be getting chance. No, no, not changing. You're from a school? No need to change, no? But you guys not changing. You have to, ne? You both have to find a school, ne? Did you apply? Go for the interviews. Maths B means you can do, okay? Right. Hi, done? Shall we write? Hi. Write number 1. Centrosomes reach the opposite poles. If you follow the diagram, you will understand nicely how it came to the poles, right? Right there, number 1. Centrosomes reach the opposite poles. Number 2. The chromosomes have arrived. The chromosomes have arrived to a place called, chromosomes have arrived to a place called metaphase plate, which is located in equal distance from each pole. Next one, the centromeres of all chromosomes, the centromeres of all chromosomes are located in the metaphase plate. Next one, at the end of this phase, each chromosome of the cell get attached to the kinetochrome microtubule, at their centromere and align at the metaphase plate. Right, now read it one more time before I erase and get ready to the next diagram. Done? Anaphase. So in the anaphase, what's going to happen is the non-kinetochore microtubules are lengthened. Kinetochore microtubules are shortened. Kinetochore microtubules are the ones which are attached to the chromosome. So when they are shortened, they are going to drag the chromosome also. So due to this force, the centromere splits into two, and what are separated now? Sister chromatids are separated. But remember very well, as long as they are separated, they are no longer called as sister chromatids. So what do we call them? Chromosomes. They are individual chromosomes. Understood now? So make sure that you will be not counted them as sister chromatids after the separation. Understood now? So here are the centrosomes. Now the chromosomes are moving to the opposite poles. They are going in a way like they don't like to go, so that there is some V shape here. Now they are away from the metaphase plate, now going towards the poles. Okay, children? Right, shall we draw? All right. So shall we write that one also? I'm reading. Microtubules attached to kinetochore. Microtubules attached to kinetochore get shortened. Get shortened. Microtubules attached to kinetochore get shortened. Next one. Non-kinetochore microtubules are lengthened. Next point. First one shortened. Next one. Non-kinetochore microtubules are lengthened. Next point, as a result, cell elongates. Next one, sister chromatids are separated. Sister chromatids are separated at the centromere. Sister chromatids are separated at the centromere. By the end of anaphase, no, one more sentence before then. Don't write by the end of anaphase. Cut that and write there. Sister chromatids are pulled towards the opposite poles. Sister chromatids are pulled towards the opposite poles. Next one, by the end of anaphase, equal and complete set of chromosomes, found at each pole of the cell, found at each pole of the cell. Found at each pole of the cell. Right, now you guys tell me what phase is coming up. Telophase. So together with telophase, usually the cytokinesis is also happening, so that they have started to show some features of cytokinesis also along with the telophase. So here, this is an animal cell now we were drawing, so it is forming a cleavage furrow, some pinch in between the cells. So we call it as cleavage furrow. So here, this is the cleavage furrow. The nuclear envelope reforms. chromosomes will decondense and they will form a network of chromatins. Nucleoli reappear, spindle and aster disappears. We can say disintegration also. So what's the name for the newly formed nuclei here? We call them as daughter nuclei. Why is that? Daughter. Mother was there, the beginning cell, the initial cell. Now here there are daughter nuclei. These are called daughter nuclei. They are genetically identical to each other and to the mother cell. Alright, start drawing. By the way, is there anyone who didn't submit plasma membrane essay? All submitted, no? Do you have it today? Uh, we are writing usually essays in the class, so I'll be marking and giving. Usually I collect all of them and mark it together, right? So if you are getting late means others are also getting their one late also, right? And what about prokaryotes, eukaryotes essay? That's a choice, right? If you don't like, you can remain without writing, right? So it's your choice, but it's a rare chance to get it marked by a marking examiner who is going to the exam, right? So usually the teachers who are doing tuition, mostly they are not going for marking, right? I'm going for marking also, so that get it done, okay? Please send me a photo of that your essay. Can you do that? Clear photo, scan image. Those who have not submitted, right? And the prokaryote eukaryote essay also, because we had to move into the new essays also after finishing that work. Give me 1 minute to continue. right there. What are the events happening here? Anaphase, telophase, nuclear envelope reforms around, nuclear envelope reforms around each set of chromosomes, each set of chromosomes at opposite poles, at opposite poles. Next one, nucleoli reappears, nucleoli reappears. Nucleoli reappears. Next one, spindle microtubules, spindle microtubules get depolymerized, get depolymerized. What's the meaning of depolymerized? They are less condensed now, okay? Depolymerized. Ah, no, not decondensed. That is kind of a broken into pieces, no? Depolymerized. Hmm. Chromosomes unwind, chromosomes unwind and chromosomes unwind and become less condensed, become less condensed to form chromatin, become less condensed to form chromatin. Right? Next one, at the end of telophase, at the end of telophase, two genetically identical, two genetically identical daughter nuclei are formed. Daughter nuclei are formed. Two genetically identical daughter nuclei are formed. And right, read it one more time. Right, done. Now the nuclear division is over. Now the cytoplasm is going to divide into two cells. With that, cellular organelles are also, subcellular components are also divided into two. They are moving to two cells. So I told that there is the cleavage furrow formed when the animal cells are dividing. So here is the cleavage furrow. From that, they will bud off two new cells. Two daughter cells are genetically identical to each other as well as to their parent cell. You can draw two separate cells as well. That's also fine. This is in an animal cell. So here is what going to happen in a plant cell. Plant cells are having cell wall. No? So this is one single cell, let's say. Here is the cell wall also. So they are going to make a structure called cell plate. This is called cell plate. Alright. So they are forming cell walls to both the sides, and then only they are making two new cells. Alright. So there's an important organelle involved in cell plate formation, that is Golgi apparatus, right? Alright. You can remember Golgi is producing things necessary for the cell wall synthesis, right? So this is how it is happening. So here having two nuclei which are genetically identical to each other, like in previous case. But don't draw centrioles, they are not available. They are in the plant cell. Understood now? Hi. Shall we draw and label? Kada putena? Right there, quickly. The division of the cytoplasm, the division of the cytoplasm starts at the end of telophase. The division of cytoplasm starts at the end of telophase. Therefore, at the end of mitosis, two genetically identical within brackets to each other and their parent cell, to each other and their parent cell. Close brackets and write daughter nuclei are formed. So daughter cells are formed. I'm sorry. Daughter cells are formed. Daughter cells are formed. In animal cells, cleavage furrow is formed. In plant cells, cell plate is formed. Cell plate is formed as a result of the vesicles produced by Golgi apparatus. So are you done? Alright. So there is some diagrams given along with the table. Please get that and paste it also. Colored diagrams are given. No, it's given for meiosis. So keep it with you. You guys can paste when it comes to that place. I'll upload it also so that the ones who are not having can get a printout and do that also. But give me a little reminder to upload that one. Alright. Now here is the challenge. I'm going to give you a table to fill without helping, right? So now please read the complete mitosis entire thing. Meanwhile, I'll put the table on the board. Don't look at the board, look at your book and completely study, okay? Alright, now copy this table. I want you to fill it with a pencil, okay? Alright, just give it a try, then I'll help you. Are there? At the beginning, I'm not going to help, so please try and do. Make sure that you follow the diagrams that we drew. The diagrams that we drew had only 3 chromosomes. You can follow it and see how many chromosomes are there, how many DNA are there in each phase. Just consider what events are happening. Is there any doubling? Is there any reduction of number of chromosomes? And see. And the ones who are coming to the class, please give the message to the others. They can join to the class and they can get the recordings also from the class. Next week, I'll be going little early. Next week is 12th, right? I'll be going little early around 6 or 5:45. I'll let you know if I'm going early. Sometimes I may give some work also until 6:30. Let's see. Finished the Done? Hi. Now just don't change anything. Once it is done, you can change. Okay? Hari. Others ready? Online students, are you done? Hari. Here, this is talking about the given cell which has 3 chromosomes. So at the G1 phase, there should be 3 chromosomes. Do you all agree? Hari. So usually at the G1 phase, the chromosomes look like network of chromatins like this. But still, even if they are loosely packed, still there are chromosomes in it, isn't it? So that this one only look like this, isn't it? So that this is having how many DNA? 1 DNA. Okay, no? So here it should be, how many chromosomes are there in human cell? 46. So here again 1 into 3, no? So here 3, here it is 46. Am I correct now? Hi. So here in the S phase, what happens here is still it look like this, but still the doubling of DNA number happens because of DNA replication at the S phase. So that number of chromosomes are not going to change. It's same, but still what's going to happen to the DNA number? Doubling. So it should be, what's the number here? 6. Here 92. Do you agree now? Hari. Don't change anything, just look at Hari. G2 phase, nothing is changing, but still this look like this. This one is the same. So at the G2 phase, 3, 6, 46, 92 is the same. Chromata phase also nothing happened. 3, 6, 46, 92 is there. Is it missing? Ah, yeah. So it's also same, ne? Chromata phase also same. Right now? So metaphase still same, no? Hi. Anaphase is the phase where there's a little change is going to happen. What's happening? Centromere splits and chromosomes are moving to the opposite pole. So number of chromosomes going to get double now, right? So here 6. Here DNA number is not changing, no? Here it is 46 into 2, 92. Here it is 92 itself. Isn't it? Am I correct? Right. In the telophase, the cell still not separated, so that the number of chromosome number, DNA number are the same. Am I correct? So it should be 6, 6, 92, 92. Same. Cytokinesis, they are going to 2 different cells. So I'm dividing this into 2. 3, 3, 3, 3, 46, 46. Here it is 46, 46. Am I correct? So you can understand that at the beginning also there were 3 chromosomes. At the end also 3 chromosomes are there. At the beginning 3 DNA was there. 3 DNAs are here. Right? So same like it here. So you can now understand what's the meaning of that. Genetically identical. Number of chromosomes are same. Number of DNA are same. And the genes which are present in that also same. Am I correct? So please fill it correctly and highlight the last line and first line. That is the place you will understand this concept properly. Okay? Done? If you're done, tell me. I can erase this one and go to the next one. By the way, hope you all got this tuned. Cell as a basic functioning unit of life. This one. Please do the questions in that, okay, at home. So there's a lot of questions that you can do, and the tables also fill everything, right? One copy is I'm having, in, if you want I can wait. Only one is there at the moment. So have one with you. Next day we'll give you another one. Okay. If you won't get a photocopy and go for the moment, I'll give a proper one. Okay. Just give me a little reminder to bring one. Okay. Right. So do you have my number? Okay. Are you done? Write the topic for the significance of mitosis. Significance of mitosis. Significance of mitosis. Number one, maintains genetic stability. Actually, this maintains genetic stability within the generation. So think now, your skin cell is removed due to aberration or something. Skin cell is having diploid nature. That means 46 chromosomes, 23 pairs of chromosomes are there. So this is a skin cell. This should divide mitotically to give another skin cell which is similar to the previous one. Still they should maintain same number of chromosomes, 46 to 46, right? Otherwise what would happen? Genetic stability is lost if it is not having 46 chromosomes. Understood now? So mitosis is helping doing that within the generation. Now understood now? And meiosis helping to keep the constant number of chromosomes from generation to another generation. Write down the first one. Number 2, for growth and development. What's the meaning of growth, Putte? An irreversible increase of dry mass. I told that volume also can be added, size also, so I'll put dry mass here. Dry mass over time. So how this is happening? You know that egg and sperm getting together to form zygote. Zygote is dividing mitotically to form embryo. Zygote is increasing the cell number little by little. Embryo is producing an individual which is having more number of cells. Zygote is having one cell. Embryo is having more cells, isn't it? Likewise, it's increasing more and more, thereby increasing the dry mass. And the development also occurs simultaneously, which are irreversible changes during the lifespan of an organism, isn't it? So all these because of mitosis. Mitosis is increasing the cell number as well as the dry mass along with that. How is it? I write that too. I'm erasing first part. Number 3, cell repair, replacement, and regeneration. Alright? When there's a wound, cells are removed out, right? So cells are shedded. So that you have to replace that with new cells. And also you have to repair also at the same time, isn't it? So this is happening there. So repair and replacement. Regeneration, where it is happening? Animals such as gecko is shedding their tail. Neither. And starfish, when their arms are cut, they can regrow. Further. So that all are happening because of mitosis. Mitosis is producing identical cells and growing their arms or growing their tail. Understood? Have you seen geckos without tail? So they are anyhow regrowing, no? Isn't it? So this is regeneration. Write these two. Hari putih. So next one is asexual reproduction. Asexual reproduction helps us to get identical individuals, isn't it? So, hari putih tira. Kamanne hari. Hari kamanne දැන් වෙන්න තමයි ඕන කොහමද වෙන්නේ. ඔතන. ආන්නේ කමන්න. හරි. Okay. Right. Identical individuals are formed. Plants also does asexual reproduction. Animals also. And fungi also doing asexual reproduction, isn't it? Many animals are showing asexual reproduction. All these cases, mitosis is the one which is helping. Can you tell me what are the asexual reproduction methods seen in plants? Vegetative propagation, very good. We can say another name for asexual reproduction in plants as vegetative propagation. There are so many vegetative propagation methods in plants. What are they? Layering, grafting, budding. Tissue culture we write separately here. What else? Stem cuttings, leaf cuttings, isn't it? Have you done these things? Very interesting, no? How many of you have done these things at home or school? No one has done? Okay. What else? Using bulbs, rhizomes, combs, those also included into this one, neither. Then separation and planting, those plantlets. Okay, no? Alright. Do you know any animal asexual reproduction methods? This hydra is showing budding. Parent hydra is there like this. There's a small bud coming up. So this is baby hydra, okay? Right. So what else? Planaria like animals, when you cut into pieces, they grow into different planaria. That is called fragmentation. And in the beehive, the male drones are formed because of parthenogenesis. These terms you will learn in lessons, right? What else? Well, yeast is also showing budding, so we can add it here. Alright, shall we write this? Good question. Asexual reproduction methods are there, but still they don't show any mitotic divisions, okay? Simple cell divisions, that's what we call. We don't say that asexual reproduction mainly, right? We tell that simple cell division is shown by bacterium, cyanobacterium, okay? There's no nucleus, no, so no mitosis. And Done. Quickly write the topic meiosis. We'll be moving to the meiosis also today. So this is the second type of nuclear division, meiosis. If you compare with mitosis, meiosis is having the number of chromosomes, right? To an individual will be producing N cells, right? So diploid to haploid. So before meiosis also, cells should get ready to divide so that there is an interface before meiosis also. Ne? Right. So interface, there are three phases, G1, S, G2. After that, in the nuclear division, there are two phases. Meiosis 1 is there, meiosis 2 is there. So in meiosis 1, these phases are there. Prophase 1. What comes next? Are we having pro-metaphase now? No, then directly metaphase 1. Next one, anaphase 1. After that, telophase 1. After that, cytokinesis. We are not going to write 1 or 2 here, but still this is the first cell division. And I'm going to meiosis 2. You should write after this one. I'll write it here. Meiosis 2. In the meiosis 2, it begins with prophase 2 and metaphase 2, anaphase 2, telophase 2, cytokinesis. And this is how it happens, right? Meiosis 2 is more or less similar to mitosis. So that we are not going to emphasize much on this section. You can just remember mitosis and write this stuff. The most important two phases in meiosis are these two. Only phases that you do have to worry about, right? So you have to study and by heart what are the events which are happening here. Other phases you can eventually write if you know mitosis. Other. Hi. So write this down quickly. I want you to do this thing also. Write the topic interface and draw a cell which is at interface and list down the things which are happening in the interface. Like in the case of mitosis, copy from that. Okay. Quickly do that. Take a little time because I can start the next part also quickly if you finish quickly. Finish? Okay. Interface G1, SG2 all done? Hi. Write the topic Prophase 1. Hi. So, like in the case of previous prophase, the network of chromatin is now going to condense and form chromosomes with two sister chromatids, right? So, here is one chromosome I will draw. Correct? So, I told that there are 46 chromosomes in a human cell now. So, at this phase, their homologous partner is going to come closer. I'll tell you what it is in a while, but I'm going to draw a similar chromosome here. Going to pair. Now 23 pairs are going to form if we consider a human cell. Right? These chromosomes we called as homologous chromosomes. Right. So, the meaning of homologous chromosomes, I'll tell you a primary definition first. At your level definition is this one. Chromosomes which are similar in size, shape, and centromere position. Well, size they are equal. Shape also they are equal. And centromere position also same. This is the basic definition. But the reality behind this, you will be learning this about this in unit 5, which is, sorry, unit 6, which is genetics, right? In that, they would say a nice thing. This is having DNA molecules, no? So, let's say there's a particular gene called capital A. Let's say these are two sister chromatids now having the same DNA molecule. So, that capital A should come to the next one also. Ne? Let's say this is eye color determination gene. We have 23 pairs of chromosome. Let's say 20th chromosome, I'm just telling a lie. 20th chromosome, eye color determination gene at this particular position. So, that is capital A. This comes from mother, so that we call this chromosome as maternal chromosome. Came in the egg cell. So, what about this one? What is this? Paternal chromosome. So, this is also 20th chromosome from father. That is 20th chromosome from mother. So that this should, at the same position, this should have a similar partner of this one, isn't it? We call them as alleles. Gene is having alternative versions, alleles. I'll explain you what it is in a little while. Anyhow, this may be having either capital A, else it can have its alternative version, simple A, simple A. Little change. You know about nucleotides now? One or two nucleotides different from the previous one. For example, capital A would be determining black eye color. Simple A would be determining blue eye color. Okay. Capital A, we call it as dominant one so that the character will come out because of capital A. So that what color would be this individual? What eye color? Black eye color. Hari? Okay. So what is hidden here is called as recessive one. You will learn that in unit 5. Don't get the deep meaning here. I'm just telling what is actually the definition here, right? So here the big definition is alleles for a particular gene is located at same location. Those are called homologous chromosomes. Understood now? Let's take another gene. Here, this position, let's say. Hair type determination gene. That will determine whether your hair is curl hair or straight hair or whatever, right? So, it comes here. Understood now? Hi. So, that's the advanced definition. Advanced definition will go when we go to unit 6. So, don't worry about that. Just remember now the chromosomes which are similar in shape, size and centromere position. Biology, similar and same are two different terms. If it is same, it should be identical. Similar means slightly similar. Okay. Hi. Almost similar, there's a slight change. That's the meaning. Hi. Okay. So now in this case, these homologous chromosomes are paired like this. Understood now? Hi. Quickly draw this one. I'm adding one little thing here. Here, these we call as sister chromatids. These two colored ones, what are they? Non-sister chromatids. Kylie, you were absent last week, right? Did you go through recording? Balana, that part is important. No? Ask if you have anything to ask, okay? Anyone else got absent last week? Did you come? හරිද පුතේ? නම් දෙක නම් මතක නෑ හොඳේ. ටික වෙලාවක් යයි මට නම් මතක තියන්න වෙලා යනවා. හරි නේද දරුවෝ? හරි. So now I'm going to tell what's going to happen next. Now these chromosomes are paired. You know that there are 23 pairs now in humans. The last one, 23rd pair is X chromosome pair, no? You know that X chromosome is bit lengthier than the Y chromosome. So remember very well, particular region of X chromosome is homologous with Y chromosome. Not every region, right? Okay, because of the size. So now special protein comes and binds this homologous tightly together. This is like keeping one over the other and binding like, okay, so this one 3D one I can't draw, so I'm drawing it like this. So keeping one over the other and binding with some glue like thingy, okay. Right. So here is what it's going to happen. This particular process we call it as synapsis. Okay. Then I'm coming to the next one. At this point, they are going to entangle their chromosomal arms. Here. Till they are attached. So what's going to happen is, at this point, they are going to break. They are going to break. They are going to exchange. So what's going to happen here is, the black color chromosome going to get part of blue color chromosome. And this one going to get part of black color chromosome. Break, exchange and rejoin. At those points, we call this process as crossing over. This is not happening only at one point. Along this length, entire length, it can happen at many places. But remember very well, it's not a must thing to happen. There may be chromosomal pairs without having crossing over. As if they are lengthier, more number of times crossing over. If they are shorter, there's lesser chance of crossing over. You know, like if there are two threads together, there's high chance of entangling if they are lengthier. If they are shorter, there's less chance. So likewise, they are also making crossing over between the non-sister chromatids. Okay? They are having the crossing over between the non-sister chromatids of homologous chromosomes. This is happening when they are lengthier. It's not a must thing also to happen. It can skip and go. But still, 23 pairs, there are higher number of crossing over happening. Okay? So, what is exchange actually? Can you understand? Here it was A, simple A, simple A. Now, here is what? Simple A is here. Capital A is here. Is it A? Am I correct? What is exchange now? Genes are exchanged. So these new chromosomes are now going to be different from the mother's one and father's one. So here is the place genetic variation occurs. Daughter chromosomes are having different combination of genes compared to the parent cell. That's the importance of meiosis compared to the mitosis. There's no change in the mitosis. Understood now? Right, now next step, what's going to happen is this synaptonemal complex breaks down. Alright? The chromosomes are slightly apart from each other, but still they are attached at the points of crossing over. Online students, if you didn't hear me, please draw this one quickly. If you don't understand anything, ask. What happened here was here the pairing and physical connection of homologous chromosomes. This is synapses, and they started to entangle their non-sister chromatids, and crossing over occurred. Crossing over means parts of the DNA molecules of non-sister chromatids, they break, exchange, and rejoin, meaning that it's forming new genetic combinations here in the new chromosomes which are going to form, which is different from the parent cell. And here now synaptonemal complex disassembles, and the chromosomes are a bit apart from each other, and the points of crossing over still there. They are called chiasmata. There may be one, two, or few chiasmata over the entire length of chromosomes. So if you are done, you can go home. Others can write and go. I'll be here. So everyone, good evening and have a nice day. So study well. You have 2 weeks for the exam paper. So please study really hard. And others who are doing online and watching the recordings later, please collect your tudes. Along with that, you will be getting your paper as well. And please don't open it and keep. Please open it on the day that you are having the paper. So it will be good for you.